So in this video, we are going to kind of check out the worksheet and also figure out how to set this simulation up to complete the worksheet. So get ready. So this is our worksheet. This is page 1, and page 1 has us using PhET, and it says our goals are to study the function of a cell membrane and the transport proteins. In the simulation, they're called channels, but they really are a form of transport protein, or what's known as a channel protein, and they look like this or this in the simulation. And so we want to learn about cell membranes and the function of the transport proteins. We also want to study the movement of solutes, or dissolved particles, across a membrane. So we want to learn about solutes, or dissolved particles. We want to learn the function of a cell membrane, and our other goal is to learn the function of transport proteins. Alright, it says get started. Google PhET membrane and run it. Well, we're already there if you've watched the first video. The next thing is to watch the video you literally are watching now. Isn't that weird? It says watch the tutorial video. Congratulations, you are currently. Alright, it says investigation 1. I'm going to help you set up investigation 1 so that you can complete the questions below. Get ready. Alright, to set up investigation number 1, first we got to add some solutes, or some dissolved atoms or molecules, or ions, depending on what the particle is. In this situation, we just have little green balls, so this is our solute. Alright, so you just tap the button. I'm going to tap it 20 times. Get ready. It's going to be loud. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. And done. Now I'm going to add some blue diamond solutes, and I'm going to add 20 of them. Are you ready? And set. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Done. Now, without channel proteins or transport proteins of any kind, these particles are never getting across this membrane. Some particles can go through our cell membranes, or some solutes can. Water does. Oxygen may, or carbon dioxide, may be able to flow right across our cell membranes, our plasma membranes, made of the phospholipids, that special type of lipid. Well, they can kind of fit through the lipid polymers that make our cell membranes. But this membrane ain't letting these particles through. And there's certainly solutes of different size or charge, if they're ions, that can't get through a cell membrane without special help from proteins. So what we're going to do is we're going to go with the green channel protein, or transporter protein, and I'm just going to put 4 in there, and we're going to observe what happens. This is what you should be doing. 1, 2, 3, and 4. You study the behavior and what's happening, and then you answer stuff on the worksheet. Now, it wouldn't be any fun if I sat there and let it play. You'd watch the video and I'd show you everything. So I want you to be able to discover for yourself, so I reset it. But again, you put 20 blue, 20 green, and then you choose the green leakage channel and you place it in. And you're supposed to do 4. 1, 2, 3, 4. Once you have 20 blue solutes and 20 green solutes above, and you have your 4 channel proteins or transporter proteins that are the green ones, you then start recording observations on the worksheet right here. So it says solute movement in and out of the cell membrane. So you describe that as best you can. I left a little space in between these 2 bullet points so that you can write underneath as well, because these lines probably aren't enough. And then it's, what's the function of the transport proteins? Pretty simple, you probably already know it, but I'd like you to describe it in the best detail you can. And then it says, how does this demonstrate selective permeability? And you may recall from slide 39, that's where the definition for it was. So if you watch this video right here, you can review, so you have to go to Google Classroom, of course, but you just review selective permeability, and you can apply that to what you saw in the simulation. So use the simulation to better help you understand the idea of selective permeability. So how did the simulation demonstrate selective permeability? And just the hint is it involves certain types of transporter proteins and the solutes in the area. And that if you put those two together and your memory of the simulation, you should be able to describe selective permeability. But again, if you're still confused, go back to that slide 39 from our video notes, and that should help. All right, let's get ready for investigation number 2. Investigation number 2, we are going to use gated channels because we just want to have a little more fun, and I'll put 4 in. This is again for investigation 2. Do 4. Doesn't really matter where you place them. You can place them evenly. Part of me always has to do that. It's up to you. All right. With the gated channels, they open or close based on you controlling it. And some proteins, like these protein transporter proteins here, they're always open. But these gated channels can be closed or open. And the cell can do this too. They can close certain transporter proteins, or they can open them. And this one, what are called gated channels in this simulation, are just that. All right, once you get those going, then you want to click the button that says Show Concentration. One of the things we want to learn from the simulation is, what is concentration? What does that mean? This is what we're going to do. We are going to take these blue diamonds and we're going to place as many as we can on this side of the membrane. Let's pretend this is the outside of the cell, and let's pretend this is the inside of the cell. But we're just going to go. Let's see how many we get. Notice, by the way, as I place these in here, this bar is going up. And this bar only exists when we're showing concentration. So there's a connection between what it's telling you here and something about concentration. So as I place more particles or solutes outside of the cell here, this bar goes up. So what does that mean about concentration? I'm going to keep adding them up, and you'll see that that bar is kind of a bar graph, but it's showing you the concentration. So as I place more and more solutes in until it won't take any more, this bar will continue to go up. And there's still room for more. Look at that. Look at it go. And it's almost, and it's done. Okay. So I've dissolved a lot of solutes outside of this cell here. There's many of them. Now, the next thing you would do to complete investigation number 2 is you would open these channels. So you'd click there, bloop, and you'd watch it for a while. You just open up the channels and you would describe basically the behavior of the solutes. How do the solutes move? Are they moving in one direction? Do they move in multiple directions? Are they moving back and forth? Do the solutes only move from outside the cell in? Do they not want to go through the channel? All these things. You would just describe this. Remember, again, we're describing this as the outside of the cell. This is the inside of the cell. So, for number 2, it said, based on your observations, what do you think concentration is? And when the gated channels were open, how did the concentration change in or out of the cell? You've seen enough at this point to be ready to do the simulation for yourself. Go ahead and get started.